Good morning, everybody. Okay, well, let's go ahead and get started this morning. A couple things. I will spend the last half hour of the class going through final exam class stuff and everything. I want to go ahead and finish up the information for the class. And as you remember, we finished off last class by looking at the functional groups that we have in organic chemistry. And I wanted just to take a little bit of time and introduce biochemistry. No, by no means am I doing anything more than an introduction. And one of the things that I want to explain to you is going to be important, and you need to think about this. If I have a carbon, okay, if I have 4 things attached to it, now I thought about using my model sets and showing you this, but they're at school. If I attach 4 things to the carbon, and if I have another group that is attached. Now, in this one, it looks like it's a square, but it's not a square. We know it is in a tetrahedron. So I'm going to redraw this, hopefully in something that you can see as a tetrahedron. If I draw this one, and this one, these two are mirror images of each other. But just like my hands are mirror images of each other. Okay, now I can put my hands together and they're like that. But if I try to put them on top of each other, notice that my hands are not superimposable. The same thing happens with molecules. That is a property we call chirality. And there are chiral molecules. And it's going to be important for biochemistry because in biochemistry, only one type of chirality is used in a lot of things. Now, I'm not going to spend a lot of time touching on it or teaching it. I just want you to know that it exists. And with that, I'm going to go ahead and do a short PowerPoint on biological molecules. And yes, trust me, it's going to be short and I'm going to go through really quickly, but this is going to be recorded, so you will be able to look at it later. And I'm going to go ahead and share a screen here in order for you to see it. So I will be talking a little bit away from this. Let's see if this can get there. All right. And maybe, just maybe, it will work. Okay. Okay, we'll see if it starts here. All right, there we go. Now, hopefully you can see that. If not, somebody chat at me that you can't see it. But let's go through. We're going to go through and introduce some of the biological molecules, proteins, carbohydrates, nucleic acids, lipid and cell membranes, and I'm going to introduce metabolism. And again, remember that all of this is just a simple introduction to it because in order to really understand this, you probably cover more of an introduction in some of the biology classes. But I want you to see how the arrangement of the molecules gives it the properties, the chemical properties that we're familiar with. So let's start with proteins. Proteins are made of amino acids, and you can read all that stuff there. I'm not going to read through everything. But what's important is that when we look at the amino acid here, now hopefully you can see my mouse here. Here we have the amino group, and over here we have the acid group. So I have 2 different functional groups on the same one. Now notice this is an acid. This oxygen is going to be very negative. So this is going to be a very acidic hydrogen, means it's easily lost. Now this amino group, I should have put it on here, but I didn't take the time to do that. This amino group has a pair of electrons here, meaning it can act as a base. And that base can actually pick up a hydrogen and become protonated. Okay, and what happens is that in an amino acid at neutral pH, like in body pH, this is going to lose a hydrogen, this is going to gain a hydrogen, and it will become something called a Zwitter ion. Now, this is what I was talking about with the chiral carbon. It's got 1, 2, 3, 4 different things attached to it. And those 4 different things that attach to it can be attached in a different way. Just be aware that all of the amino acids are the same kind of chirality, and that you will need to study that at some point. Okay, each amino acid has a 3-letter abbreviation. I don't like this slide, but I didn't go through and edit it. Every amino acid has a name, a 3-letter abbreviation, and a 1-letter abbreviation. This R group is different for all of them. If you put a methyl group there, a CH3, this becomes alanine. Probably my favorite amino acid. If you can't figure out why, that's okay. You need to drink coffee then. All right. Here we have the 20 most common amino acids: serine, threonine, cysteine, tyrosine, asparagine, glutamine, histidine, arginine, lysine, glutamic acid, aspartic acid, glycine, methionine, alanine. See, there it is. That's mine. Phenylalanine, valine, tryptophan. That happened the other day. There was one lying in the middle of the floor, and I tripped right over it. Okay. Leucine, proline, isoleucine. Now, I'm not going to spend a lot of time on these. I just want you to be aware that they exist. Notice that I have the amino acids in here drawn in the ionic form, where this is deprotonated and that is protonated. This is called a Zwitter ion. So when you look at this, this is like an extremely polarized thing. And you're wondering, okay, so these are the amino acids. I know they're not very nice. Okay. All right. I will still tell jokes whether you guys laugh or not. Okay. All right. So these Zwitter ions are dipolar, but there's no net charge to them. They form a link called a peptide bond. So if I put 2 together, if I put an amino and an acid together, and I'll draw that out here in just a couple minutes, we actually have a condensation occurring. Okay. All right, let's see. And if I put a bunch of them together, I get a polypeptide, and these amino acids come together to form things that act. Some of the things they form are like hemoglobin or enzymes. Okay, hemoglobin is one that's pretty important. It's the molecule in red blood cells that carries oxygen from the lung to all of the body's other cells. And it's got 4 different big chains called subunits. And they hold the subunits together by hydrophobic forces. In other words, it's not hydrogen bonding that pulls it together. Okay. Here we see the heme part of the hemoglobin. This is really important. And just to remind you, remember I said that these metal ions, these transition metal ions, play really important roles, even though they're a very small amount. Without that iron in this heme group, it doesn't carry oxygen. So that's really important. Not going to spend a lot of time on it. I just want you to know that these exist. This is a central portion of it, and at the end of each of these is a chain of amino acids that holds it into its 3-dimensional structure. So we see the primary structure is just the order of amino acids that are put together, and notice the acid end is connected to the amino end of one, and that continues on. Okay, we have a secondary structure. They can form helices, they can form sheets. Those are known as secondary structures. A tertiary structure is the 3-dimensional shape, and if I have more than one unit, I get a quaternary structure. Again, just introducing this, hemoglobin was a nice one to do that. Enzymes. We spent a lot of time in this class studying catalysts, and enzymes very often are catalysts. They serve as biological catalysts. What they're really good at is taking things that wouldn't normally collide with the proper orientation, like maybe 1 in a million collisions are going to have the proper orientation, and putting them in the proper place to collide correctly. And often that collision is not energetically favored, but by the use of an ATP, they can cause a reaction to occur by adding energy to it that wouldn't normally happen. There's a number of other things that enzymes do. I'm not going to spend a lot of time on it, but this group of proteins forms enzymes. You can recognize the name of the enzyme because it ends in ASE. Okay, like a polymerase or a helicase or anything with the ASE should be an enzyme. Enzymes have active sites, which is where this reaction is going to be occurring. An example is lysozyme, which I'm not going to go through, but it's a protein containing 129 amino acids in a polypeptide chain. This is the primary structure. This is just the order of the amino acids that are involved in it. Okay. And notice we have the acid end to it and the amino end to it. But this is not what it looks like. Now, the cysteines both have sulfurs and that form something called a disulfide bond. This gives it a brace to the structure. It allows it to do that. You also have other things. There are things like phenyl groups where the benzene rings will help attract to each other, or you have some where the side chains are polar. You have a positive polar and a negative polar one can attract to each other. These all hold this together into another shape. And here we see a picture of the lysozyme in its three-dimensional shape that we see there. Okay. All right. Next thing is carbohydrates, also known as sugars. They give energy. I'm not going to go through all that slide. A monosaccharide is a single polyhydroxyaldehyde or ketone unit. And if we remember, an aldehyde is a terminal carbonyl and a ketone is a carbonyl in the middle of a chain. A carbonyl is a carbon double bond to an oxygen. If I have two of these units, it's a disaccharide. If I have a bunch of them, it's a polysaccharide. And I just want to look at this. This is what is called a Fischer projection. This is one of the things that we see often with glucose. And fructose. Yes, that is correctly pronounced fructose. A couple things that I want to point out. First of all, glucose. Notice that the carbonyl is on the end. This is an aldehyde, so it's known as an aldo. And it's got 1, 2, 3, 4, 5, 6. So this is an aldohexose. Now remember, I said an enzyme ends in ASE. If it ends in OSE, it's a sugar, like glucose or fructose. Okay. And notice it's got a lot of hydroxys on there, means it's very soluble in water. Here we see fructose. Fructose is 1, 2, 3, 4, 5, 6. It is also a hexose, but it is a keto sugar and not an aldo sugar. So it's got an aldehyde for the glucose, a ketone for the fructose. So it's known as a ketohexose. In addition, there is some chirality about the linkages, whether the oxygen is in the point going down or the point going to the side makes a difference. In sucrose, we have an alpha linkage, and in lactose, we have a beta linkage. Now, it's interesting. There are a couple things, like, for example, glycogen. Glycogen is just a big string of glucose, and it can be broken down easily. But cellulose is also pure glucose, but we can't break it down. And the only difference is between an alpha and a beta linkage that we have there. Again, you can study that more. Here we have amylose, also known as starch. It's got the alpha linkage. Cellulose has the beta linkage. So then we get to nucleic acids. I have just put these slides together because I want to show you a couple things about nucleic acid. I thought about editing them, but we're not going to go through that. The different DNA and RNA structures are put together by, there's a three-part to it. You have your base. It's a base because it has that amine group in here. That amine group has a lone pair of electrons, which acts as a base. This particular one is adenine. Okay, it is attached to a nucleic acid, and it's a ribose, okay? Now, this particular one is a deoxyribose. This is just a ribose. Notice the OH here and just the H there. So if I take an oxygen off of there, this gives it a more stable structure. If I've got the OHs here, those OHs kind of destabilize the structure a little bit. This hydrogen here makes this a much more stable and lasting structure. That's why it is. And you've probably seen adenine, cytosine, guanine, and thymine. What's important is how they're put together, okay? Now here is another picture of the ribose and the deoxyribose. Okay. Here we see the structure, and this is the complementary structure. I have two strands of DNA, and they're attached to each other through hydrogen bonding. All the A's are bonded to T's, and all the C's are bonded to G's, but they're all bonded through a series of hydrogen bonds that we see here, okay? And that always works out that way. All right. Let's get this working again. It's not advancing on me. Give me a second here. There. Now, one of the things I want to point out is the helix was deduced by Watson and Crick, and Watson and Crick actually got the Nobel Prize for this. But it was based on X-ray diffraction data from Rosalind Franklin. And without Rosalind Franklin, Watson and Crick do nothing. Now, Watson and Crick received the Nobel Prize for it. Rosalind Franklin did not. Now, I will tell you right now, the reason that she was not given was because she had deceased. She was deceased when this particular Nobel Prize was given out. But if you talk about Watson and Crick without talking about Rosalind Franklin, you are ignoring a significant part of history. Now, in case you're wondering, Rosalind Franklin was in inorganic chemistry. She was working with X-ray diffraction and is one of the pioneers of X-ray diffraction. Very important person in chemical and biological history, and why her name is not on the double helix model is something that still bothers me today. Okay. Now, the double helix is a secondary structure. I'm not going to go through all of that. You'll spend a lot of your time. I did want to point out the contribution of Rosalind Franklin, and you should know that name, even though I'm not going to put it on the test. Whoops. Then we get to protein synthesis. I'm not going to go through all this. I just figured I'd leave these slides and go through messenger RNA, ribosomal RNA, transfer RNA. You can look through all of those. What is important to know is that that series of bases, adenines, thymines, guanines, cytosines, all of those, if I put a particular sequence, those can be read, and that information codes for a particular amino acid. Now, what's interesting is that you could have one code that starts on one place in the nucleic acid, another one that starts one nucleic acid over, and you're going to get an entire different code. So the amount of information stored in DNA is phenomenal. In case you're wondering, for comparison, for computers, we have a system that only has two possibilities, a 1 and a 0, and all of our computer data is contained in that. For DNA, we have 4 different possibilities. So take the amount of information squared versus those numbers, 1s and 0s. You can have a huge amount of information. And then add to that, that if you just move over 1, you get an entire new set of information. The amount of information in DNA is incredible. It is large. We do not understand all of the information that's there. And probably in your lifetime, by the end of it, I really doubt we'll still understand everything that's there. Although in my lifetime, we've made huge, huge jumps. The Human Genome Project, for example, where we have a better idea of where everything is. These are just examples here. The transfer RNA, I put that on there because it shows the attachment site to the amino acid. And then we can see the protein synthesis. I'm not going to go through that. I do want to introduce lipids and cell membranes. Fatty acids are, notice the acid to it. Now, if I take an acid and an alcohol together, I get an ester. So this is really an ester. So this is a glycerol. Uh, it has 3 hydroxides on it, and each of those hydroxide bonds to an acid, giving me an ester. So this is known as a triacylglycerol or triglyceride. The fatty acids are carbon chains. So basically they have the acid group with a whole bunch of carbon chains behind them. We give names for them. Lauric acid is a carbon 12. And yes, you will find this if you read the ingredients on, say, shampoo. You'll see a laurate. Remember, if I take an acid and take the hydrogen off, it becomes an ion. So if I take acetic acid, remove the hydrogen, I get acetate. If I take lauric acid, I remove the hydrogen off of there, I get laurate. Okay, and those leave me the ions. Okay. Number of different acids that you see there. Now notice that for some reason or another, an even number of carbons tends to be more prominent in that. We don't see any with an odd number of carbons. That's true. I'm not going to go through the reason of that right now, even though I'm not 100% sure that's known. But it just so happens that the common fatty acids have an even number of carbons. Okay. Then we have steroids. Steroids are a base of compounds. And I always tell people, if you're looking for a steroid, look for the duck. See the duck bill here, the head, the feet. Look for the duck for a steroid. Cholesterol is a steroid synthesized by liver. Phospholipids are another one. And notice that instead of a glycerol there, they also have a phosphate attached to it. And in this particular one, it's a choline, but it's got a phospholipid. These are a little bit different. Now notice they have 2 fatty acid tails there. And if I put them together, I get the phospholipid bilayer. And you can see the polar head and the nonpolar head. I'm not going to go through all of this, but I do want to point out that the nonpolar tails are attracted to each other. The polar head is attracted to each other. So this sits in an aqueous solution quite well without the water penetrating the cell through that. And here is the normal fluid mosaic model. Metabolism, I'm not going to go through all of this. I do want to point out the adenosine triphosphate. Now notice that this is the same adenosine that we see in DNA. Okay, and by adding a phosphate to it, I can store energy. By removing a phosphate, I can release energy, and that is the basis of metabolism. And I know that was a really quick, short PowerPoint presentation. And I am going to step back up here. Now, by no means did I cover biochemistry. I would say at best today, I introduced biochemistry. Now, there's a couple reactions that I did want to show you. If I have an amino acid, I have to put alanine up there because it's my favorite one. Okay. If I have 2 amino acids together, and I put another one here, oops. Helps if I draw it right. I accidentally put the methyl group here. It does not go here. This already has 1, 2, 3, 4 bonds. It has to go on the alpha carbon, which is the same one our amine is attached to. Now, notice in this case, now I'm going to do something here. I'm going to redraw this to where it's got a hydrogen here and a hydrogen here. And I'm going to redraw this as an OH. Now notice, I have 2 hydrogens and an oxygen. And now you're thinking, what are you going to do with that? If I remove that, I get a bond between here, and I'm just going to remove that here and draw that big bond there. This reaction is called a condensation reaction. It's very important. A lot of the polymers that we have are formed by this. Now, most of the biological polymers are formed by condensation reactions. Just on a quick side note, the plastics that we have, there are some that are formed this way. Most of them, they have a double bond, and that double bond is broken to add another structure. So for example, I take an ethylene and I break that double bond and I add another ethylene there. That gives me polyethylene. Okay. All right. Wow. I have managed to cover everything. I would tell you I covered everything well, but I'm not sure that's correct. I do want to spend a bit of time in preparing for the final exam and a little bit of time finishing up the class. Okay, for the final exam, I haven't decided the number of questions. It is either going to be 60 or 70, not any other option. I am going to have you plan on 110 minutes for the exam. So you will have an hour and 50 minutes for the exam. The exam will be open at 10 o'clock on Monday. I will have it require a lockdown browser. I am not going to require you to use the Respondus. I probably should do that, but it's going to be simpler to me if I just require the lockdown browser. Don't tell the ACS this. Well, maybe they will find out, so I will do that. But there are a number of students who can use the lockdown browser that are not able to use the Respondus system. So we're going to go with the lockdown browser. If you cannot use a lockdown browser, I need you to contact me. If you can find a way to use that, that is going to make life a lot simpler for me and better for you, but we'll figure it out one way or the other. With the lockdown browser, you are going to be able to open the questions. They will be visible there, and then they're gone. That's the big thing. The ACS wants them secure. Although I will tell you right now, they're probably not going to be that secure. Don't spend your time trying to figure out where you can find these questions. Worry about studying for the final exam. What do you need to do? Well, we're going to review a little bit. Now, tomorrow I am going to have some office hours from 11 to 11:30 to 11:45. I haven't figured out exactly how long that one's going to go. So I'll have some office hours at 11, and then from probably 2 to 2:45. So I will send an email out with that once I verify that those are the times that I will be available. I am 100% sure about the morning time. The afternoon time, I have a meeting at 1 o'clock that is supposed to be done by 2 o'clock. So I can't promise I'll have it open right up at 2. But it will be within a couple minutes of 2 o'clock when I open those up. I was debating about whether to do a review session on Friday at 10 o'clock. I have a meeting that I'm told is not optional. I have 2 other meetings that day, so I don't have time to put a live review session. My goal is to do a couple small videos of review from some topics from Gen Chem 1, and you have the videos that we have from here available on Canvas. I do want to do at least a little short review of colligative properties, kinetics, and a little review on equilibrium. I will try to get those up by the end of the day tomorrow. So there will be a couple little review sessions, but I won't have them live, so you won't be able to answer questions. I will tell you right now, I will put those videos up on the course videos page, and then I should be able to make it to where you can post questions below that. If not, I will take any emailed question you have and post it with that and answer it at that point. So hopefully that is going to help you prepare. Now, as far as grading goes, I am still working on grading the take-home exams. I think I have most of the other things graded. I have the 2 paper assignments graded, but I don't have them entered yet. Those will be entered. I will get your take-home exams graded by the end of the day Friday, and your Canvas grade by midnight on Friday will be accurate with everything except for the final exam. Remember, the final exam is 20% of your score. I will have it set that you will know your final exam score as soon as you submit that. On the final exam on Monday, I will be available. If you have technical trouble, don't wait 10, 15 minutes before you contact me. Let me know immediately when I can do something about it. If your internet shuts down or whatever, okay, I know this is going to happen at some point. Hopefully it won't be in my class. Hopefully it'll be in somebody else's, but that does happen. If that happens, contact me. Now, I think all of you have a phone that is capable of contacting me. I would prefer email, but I will send out, I think in one of the communications, I made my cell phone available. I don't like using my cell phone to communicate. First of all, it's getting old and the battery dies on a regular basis. And so, the chances of me having my cell phone dead at a particular time are rather high. So I don't like to rely on my cell phone for communication. I like email because if you email me when it's off, I have other means of checking that. So I would prefer an email, but if you have to call, there will be a number that you'll be able to call and reach me. Again, you're using the lockdown browser. All right, so with that, we have covered all of the topics in general chemistry over the year that you need to know, okay? Remember that general chemistry is an introductory course, so we have not mastered all of them. A lot of the things we said is if we simplify it down to a specific set of cases where we can limit the number of variables, we have the ability to do calculations. Some of the upper level classes go through, all right, so what if we can't simplify everything? What if we have to take a real system and analyze it? What are we going to do? So you can get into some fun things. I did want to introduce a couple of the upper division classes that I think are really important and fun. Analytical chemistry. Now, analytical chemistry answers 2 questions. What's there and how much of it is there? Okay, that's really what analytical chemistry does. If you want to know what it is or how much is there, so you have some water, you want to know what's in the water. That's analytical chemistry. You want to know how much of whatever is in the water. That's also analytical chemistry. Now, again, that's an overly simplified statement of analytical chemistry, but it's really a basis of what is going on there. And there's 2 ways you analyze things. You can use wet chemistry, and there's also various forms of spectroscopy. In between analytical 1 and 2, they cover that. I already introduced organic chemistry. Organic chemistry covers the chemistry of carbon. And trust me when I say I introduced that. I didn't talk anything about how any of these molecules react, just a little bit about their structure. And that's organic chemistry. Organic, analytical. Inorganic chemistry is like organic chemistry, but we throw metals in there, okay? We start looking at d orbitals. In organic chemistry, you're only going to look basically at sp3, sp2, and sp hybrid carbons. That's pretty much the summation of the chemistry that you're going to be looking at. Now, there's a lot. There's a lot there that can be done with just that. In inorganic chemistry, we throw metals in there. Biochemistry deals with molecules that are important to life, to biology. Okay, so that to a lot of people is the most fun. To me, biochemistry is really fun, especially when we start looking at enzymes that have those fun metals in there. That's the area that I like the most, is the inorganic chemistry, which is not surprising. I'm an inorganic chemist myself. And that's the area that I like working with the most. There's a lot of fun things that are involved with that. Physical chemistry, very important, deals with the energy involved in chemical reactions. So those are all the different areas that we have. yes, you can break chemistry down. You can talk about polymer chemistry, materials chemistry, all of that. But basically, the 5 areas of chemistry are what you're going to be looking at. And in case you're wondering, if you're going into medicine, what's the most important? Biochemistry, of course. However, do you really understand biochemistry if you don't understand organic chemistry, that is the, the basis of it? No. Are you going to understand the the system involved in blood and the blood buffers without understanding some equilibrium chemistry? No. So again, this is something that applies to a number of different areas. If you're going into medicine, pharmacy, any of those, it's really important for you to know that. Okay, so don't forget the stuff that you learned in general chemistry. And here's a bit of advice. And I know monetary things are important, but one of my recommendations for you is to keep your general chemistry book. And if you don't have a physical copy of the general chemistry book and you've been using a digital one, okay, you'll have to deal with that. But if you can pick up a cheap copy of the Gen Chem book that we have, it's going to be the best resource for you to study for a test, such as the MCAT, the PCAT, the OET, whatever test it is you're going to take. The book that you learn general chemistry from is the best book for you to use to study when you're trying to go back through. Let's see. All right. Final exam is going to be not pure percentage. I am going to have to do some statistical analysis before I figure out what percentage grade that is going to be. So I will not have an instantaneous percentage grade that your final exam will be calculated. Suffice it to say, a 50% on there would normally be an F. However, if you're at the 50th percentile, that should be a C, average. C is average. And I would expect the 50th percentile, hopefully, if it's 70 questions, hopefully you're going to be able to do in the 40s to get at the 50th percentile. But I don't know. I haven't put this particular set of questions together before and given it, so we'll take a look at that. I have an idea that a 40 is probably going to end up being a B. 40 out of 70 will probably end up being the lowest B, but that's a guess. Is the final open note an open book? No. I expect you to sit there. I will have you have scratch paper, and maybe I should do the respondus. Okay, I'm going to do this. For those that are capable of it, I'm going to have you do a respondus. So you're going to show, and I don't care if you show your ID and all that stuff, but if it makes you do that, just for test security and everything. But when it says, check your work area and everything like that, I want you to show me your scratch paper that you're going to use at the beginning of it. And I'm not going to spend a lot of time going through the respondus monitor or anything like that. The papers that you can have out, the periodic table that I gave you, you will also be able to have available there is a table, a periodic table, and equation sheet or constant sheet that the ACS also provides. I will send out those, and I hopefully will have 1 page of the test or 2 pages of the test will have those actually on the test itself, so you can scroll back and refer to that. But I'm okay if you also have printed copies of those. So if you want to have printed copies of those and paper out, that's fine. There are going to be calculations. You will need to do that. The only thing that I ask is that afterwards you take that paper and you throw it away. And this is the concern they have with the security of the test, is not so much, are people going to look up stuff and everything as they're taking the test. They're more concerned with, here is one person taking the test at this time, and they're going to scan their scratch paper and post it for other people to have. That's what they're trying to avoid, and I agree that we should try to avoid that. You are going to be one of the first groups taking this exam in the country. So don't send the information out. Okay, just take and get rid of them. All right. I still have some time. Is that a... Okay. Do we have questions or particular things that you would like me to review? I have about 5-10 minutes if we want to do that. So I will look at questions. In the meantime, I do want to say goodbye to this class. I really missed seeing you in person. I will tell you right now that you've been a really good group to work with. I will probably say out of all of the classes I've taught at Newman, this is going to be my favorite class to have worked with, possibly because of the heartache that we had with being separated and everything like that. But also, I really appreciate how the majority of you have really worked to understand what's going on. And that's very impressive. I encourage you all to continue with what you're doing. Now, if you have decided going through this, the science isn't for you, I will be sad. But hopefully you know that. For most of you, I think you know that for your goal, you know, chemistry may be something you really enjoyed, or it may be, hey, I can do this. This was my obstacle I need to get where I'm going to be. I know for some reason or another, a lot of physical therapists, this tends to be one of the big obstacles they have. You guys have done well. All right. Let's see. One more thing. Grades. I will have your final exam graded. There are some people who do not have some things turned in. At this point in time, with the way this semester have gone, I will give credit to anyone who turns in anything that hasn't been turned in at this point. If it's a homework assignment, if you missed a quiz, contact me. I will give you a makeup quiz. It won't be the same one everybody else took, but I will give you a makeup quiz. If you did not get your take-home exam in, and that might be true of one or two people, and you're wanting to pass this class, get it turned in, okay? I will have, again, all the grades calculated and accurate on Canvas by the end of the day on Friday, which means that I will be up late tonight grading take-home exams. I've graded part of them. I did get through and graded all the equilibrium section of the take-home exam, and I made comments like, you know, and I will tell you right now, because I originally said 10 points being added to exam 2, and then after the first couple of them, I'm like, I did 10 points each question. I graded fairly harshly on those. However, those were points that were added to an exam, so I don't feel bad about how harshly I graded those. That's going to be important. I will have, let's see, one other thing about the take-home exam. I do not plan on putting comments or anything on there. I am going to do this the most rapid grading that I can, which means I just am going to go through and I'm going to count points that you miss, and I'm going to put the final score on there. I am not going to go through and give a lot of feedback on it, and I'm going to apologize for that at this point. But at the same time, if you want to have good feedback and everything, it's not going to be until the end of the next week that you see anything. So I think it's going to be more important for you to see what your score is than it is for me to be able to go through everything. A lot of the final exam is going to be out of Gen Chem 2. Let me spend a couple minutes here and go through the areas that you need to know for the final exam. So out of Gen Chem 1, okay, nomenclature, naming things, stoichiometry is going to be important. Atomic structure, both how we know the atomic structure, and we know about the nucleus, We know about things like effective nuclear charge and how that affects ionization energy, all of those. A little bit of light, how we know that light, from light, how we know the atomic structure, a number of things about that. Let me just Lewis structures and everything that we learned about Lewis structures. Now, I say this, these are things you probably should review. You should review how to draw a Lewis structure and information we know from that, such as what is the shape of this molecule, things like that. Now, that's also going to be good to review for those going into organic chemistry anyway. That, gas laws, those are all things that you should review. Now, I didn't mention the thermodynamics because we really covered the thermodynamics in Gen Chem 2. In Gen Chem 2, the things that we covered, we covered colligative properties, and in general, colligative properties and properties of solutions. So colligative properties, chemical kinetics, rates of reactions, equilibria, which is the plural of equilibrium, and very specific acid, base, and solubility, equilibria, both of those were important. We also have thermodynamics, very specifically. Enthalpy, entropy, and free energy, those. And then we have nuclear chemistry, and along with this, the chapter 22, the coordination complexes, that's also important. But these are the things that I would review quite extensively. I would spend a little bit of time on that. Again, I will try to get a simple review session done for each of these. Forgive me if they're only a few minutes long, but I want to give you a chance to just review some things. I don't know at this point whether I'm going to try to do those as a single video with a series of steps or a series of videos to review. I don't know for sure which way I'm going to do that. All right. So do we have any other questions that you really want to ask me before we end the class? Okay, I see it's 10:48. I do want to do one thing at the very end of class, and that is I want to end this with a prayer. So if you would please join me in the name of the Father, and the Son, and the Holy Spirit. Amen. Dear Heavenly Father, we thank you for allowing us to learn through this time. We thank you for the gift of health, and we say a special prayer during this time for all those who have suffered from the coronavirus. We say a special prayer for all those who have died. Please bless them and their families. And I ask you to help us to do our best. Most of all, Lord, help us to do your will. And as we search and work on what we're going to do with our lives, please help us to follow your will. For those going into medicine, help us to be great practitioners of medicine. For those going into other fields, help us to do the best that we can. And together we pray. Amen. With that, goodbye, everyone. Have a great time. Please visit me next fall. Come talk to me. Come see me. I really do miss you. Bye.